well, I've already been introduced. I'm Joe Hamilton. I work with Sarah Tabrizi at University College London, where I'm a PhD student. And today I'll be presenting the gene FAN1 and why we're interested in it within the context of Huntington's disease. And it codes for, as Darren and Jill have mentioned, it's a DNA repair protein which have recently shown to modify, thank you, Huntington's age of onset. So I'll first start by outlining the function of FAN1 in an endogenous environment. And then I'll go on to discuss specific papers that have been published in the past couple of years. And these delve more into the precise mechanism of FAN1 in modifying HD. So I won't show the graph because it's been showed a number of times, but we know that the biggest modifier of age of onset is the length of the CG repeat itself within the Huntington gene. But we're now beginning to understand the contribution from other factors, other genetic factors, and this kind of comes from the seminal GWAS study from a few years ago. And this kind of outlined many genes that fell under the umbrella of DNA repair, and many specifically from the mismatch repair pathway. So we have MLH1 and MSH3, which kind of, we know drives somatic expansion of the CG repeat itself. However, what's quite clear from this is FAN1. So at the chromosome 15 locus, we see this huge signal in a continuous phenotype analysis as well as a dichotomous analysis, and this has been replicated in several extensions of this original study. And this has actually led to multiple karyotypes, which Darren also mentioned, flanking the FAN1 locus at chromosome 15, and these are believed to modify age of onset through their effects on the FAN1 gene, either by modifying expression or by coding mutations which change the function of the protein itself. And the magnitude of this has quite big therapeutic potential. For instance, the 15AM1 haplotype hastens onset by about 5 years in patients. So the therapeutic potential is also quite wide-ranging, so just it's quite pertinent to mention that variation within the FAN1 gene has also been linked with the age of onset and pathogenesis of other diseases, trinucleotide diseases such as the spinocerebellar ataxias, as well as Fragile X disorders, and also interestingly within schizophrenia, autism and bipolar disorders, which themselves have shown to contain expanded repeat loci within the genome. So what is FAN1 and what does it do? It is a 5' to 3' exonuclease and endonuclease, and it's canonically involved in the resolution of interstrand crosslinks. So essentially, these are covalent bonds that occur between DNA strands, and this impedes the bidirectional progression of polymerases and associated DNA repair machinery, and this prevents replication and transcription, eventually leading to cell death. So FAN1's involved in removing these, and it's got quite broad substrate specificity as a nuclease, so it can target 5' and 3' flap containing DNA, D-loop structures, and quite interestingly in the context of HD, trinucleotide repeat containing slip-out structures that contain CAG or CTG repeats. It's been implicated in the MLH1 interactome, so this goes back as far as the gene's discovery. It was shown to interact at the protein level with MLH1. And this forms part of the mechanistic data that we'll discuss later on from our lab and also another lab because of the relevance of MLH1 in driving somatic expansion. And the reason we're interested in FAN1 itself as a genetic modifier and the way we think it acts is through its quite clear effect on somatic expansion phenotype. So whereas the genes such as MSH3 and MLH1 within the mismatch repair pathway are known to drive expansion, we know quite clearly that FAN1 suppresses this. So this is data from back in 2019 from our lab, and we showed in this stem cell model, so it's a 109 CAG HD patient-derived stem cell model, we showed that FAN1 knockdown via shRNA treatment actually accelerated somatic expansion. And this was true also when differentiated into neurons, showing that it's not a cell-type specific effect. So this has since been replicated in other stem cell models that are prone to expansion, as well as mouse models. And it's shown to rely on mismatch repair activity, or MLH1 expression. So this is the Q111 HD mouse model, and you can see in the left column, so if you knocked out FAN1, you get these huge expansion events that are in disease-relevant tissues such as the striatum and liver. However, this effect is negated when you co-genetically ablate MLH1 and therefore inhibit mismatch repair pathway, suggesting that, well, showing that the FAN1 potentiating effect on somatic expansion relies on MLH1 expression. So it's quite clear, obviously, that FAN1 suppresses expansion, but what is the precise mechanism? Because that's what we really need to figure out. And in the past 2 years specifically, there's been quite a lot of research and data published regarding this. Most of it that I've illustrated here, a few of them I'm going to talk about in detail, and this is all from Huntington's disease, but quite encouragingly, this has kind of been corroborated by evidence from a specific Fragile X mouse model as well. So in a completely unbiased manner, I'm going to start with a paper from my lab. So this was published last year, and we basically used the jumping off point of the FAN1-MLH1 interaction and investigated its relevance to instability or somatic expansion. So we performed a structure function study in FAN1 knockout U2S osteosarcoma cells, and we reconstituted the cells with truncated or mutated FAN1 variants, as well as transduced them with a Huntington exon 1 construct harboring 118 CAGs, so we could monitor expansion on different FAN1 backgrounds. So through this work, as well as an evolutionary conservation analysis, we discovered what we call the SPIF motif in the N-terminus of FAN1 that mediates MLH1 binding. So although we call it the SPIF, I'm going to try and refer to it as the MIP box, which is an MLH1 interacting protein box, and this similarly is found in MLH1 interactors such as MSH3 and EXO1, which also participate in the mismatch repair network. So we showed that if you mutate the conserved residues within this MIP box, the SY or the F, you almost completely ablate MLH1 interaction, and it's quite strong in the full-length expressing protein that one single substitution can do this. And of relevance to somatic expansion phenotype, once you prevent the MLH1 interaction, you actually increase the rate of somatic expansion, which is shown by the latter three bars in this graph here. So it accelerates expansion, suggesting that both of the phenomena were mechanistically linked. I don't have enough time to go into the precise mechanistic data from our paper, but essentially we were able to show that FAN1 competes with MSH3 of the mismatch repair network for MLH1 binding, and that if you overexpress FAN1 and to bind MLH1, you can sequester it away from the DNA and away from the mismatch repair network, suppressing expansion and instability. However, you'll note that once you prevent the MLH1 interaction, you produce an intermediate phenotype. So it doesn't reach that of a FAN1 knockout, it's about halfway between. So this suggested to us another mechanism was still operational that was dependent on FAN1 expression. We therefore turned to FAN1's canonical function as a nuclease. So within the nuclease domain, it contains this D960 residue, and this is involved in coordination of metal ions and basically mediates catalysis. So we found a D960A mutation which negates nuclease activity, and when that's done on top of the MLH1 binding dead mutant, you get an expansion phenotype equivalent to FAN1 knockouts, suggesting that both mechanisms are sufficient to account for the entirety of FAN1's protective effect on somatic expansion. So this interaction was actually co-characterized by Alex Sartori's DNA repair group, and they investigated the relevance to repeat activity, the ability of FAN1 to resolve CAG repeat-containing loopouts. So I'll swiftly go through this, but they were able to show that the S126 residue is phosphorylated, and this actually regulates the FAN1-MLH1 complex interaction. And they actually identified some kinases that were responsible, including the proline-directed kinases CDK2 or CDK5 in particular that we're interested in. They also looked downstream of the MIP box and discovered a MIM box, which is MLH1 interacting motif box. And this is a pentapeptide motif which also acts in concert with the MIP box to stabilize the FAN1-MLH1 complex. And interestingly, FAN1 under normal circumstances can resolve these loopouts containing 20 CG repeats. However, if you prevent the MLH1 interaction, it reduces repair efficiency. The nuclease activity itself of FAN1 was recently shown quite convincingly by a group in Cardiff led by Tom Massey. So they used the 109 CAG stem cell model I mentioned earlier and performed the nuclease inactivating mutations, generating heterozygous and homozygous clones. And as you can see, with incorporation of this mutation, you actually have a stepwise increase in somatic expansion, with the homozygous clones looking exactly like knockout. So from this, they concluded that the nuclease activity could be the downstream mechanism mediating the entirety of FAN1's protective effect. And this is obviously going to need to replicate in a neuronal system as well. They provided some really interesting genetic data. So I've highlighted in red here several mutations found in the nuclease domain of FAN1, and these were consistently associated with individuals with earlier age of onset of the disease and a more severe phenotype, linking FAN1 nuclease activity directly to its genetic modifier effect. And they took these SNPs forward and clustered in the bottom left of this graph here. These are the HD age of onset hastening SNPs, and they all impaired FAN1's ability to act as a nuclease at 5' flap containing DNA by about 50% or more less function than the wild type. And finally, the last paper I'll talk about comes from Toronto, from Chris Pearson's group, and they look specifically at FAN1 exonuclease activity and describe the dynamics of FAN1 on repeat DNA. So through this, they purified FAN1 protein and did an in vitro assay with this CAG loop-out containing substrate and showed that exonucleolytically digested between CA and AG nucleotides, but not between the C and G. And this isn't one long digestive process to remove the loop out. It's actually a sequential and circular process of binding, cleavage, and then dissociation. And on this gel here, you can see these darker bands that are towards the 3' of the CAG loop out. These are what they refer to as hot spots. So this is where FAN1 seems to pause in the DNA, and it's a specific issue with DNA reassociation. And they propose that this provides opportunities for intermediary events that could be relevant to expansion or even contraction, such as refolding of the DNA into alternative structures or the recruitment and activity of other DNA repair enzymes. So just to conclude, the genetic evidence ever since the original GWAS has quite convincingly shown that FAN1 is a powerful genetic modifier of Huntington's disease, and it likely exerts this effect through its protective efficiency in terms of somatic expansion. The papers I've discussed today have reviewed two distinct mechanisms in how FAN1 mediates somatic stability, involving binding of the MLH1 protein and sequestration, potentially from the mismatch repair network, as well as directly involving nuclease activity of FAN1. But obviously there's a huge amount we still don't actually know about how FAN1 works in vivo, and some future work is aiming to look at the therapeutic tractability of targeting FAN1-dependent mechanisms. One of them I briefly mentioned, identified by Alex Sartori's group, was that the serine 126 residue is phosphorylated, and this regulates the FAN1 MLH1 complex. So we are now pursuing CDK5 ASOs to look at regulating this interaction within our 125 CAG stem cell and neuronal model. But also, finally, we really need to understand more how FAN1 nuclease activity works in the context of CAG repeat repair. So we need to look at CAG repeat structures from small loop-outs to large loop-outs, because we don't know exactly what it's targeting in vivo, and also to look at this within the context of mismatch repair. So Alex Sartori's paper suggested that MLH1 binding to FAN1 moderates its activity and interaction. So we really need to investigate FAN1 nuclease activity in this more endogenous context of functional mismatch repair. So just to finish, thank everyone in the lab involved in the project, as well as the woman herself, Sarah Tabrizi, and several of our collaborators from our paper, all of our funders that made all this work possible, and a special mention to Tom Vode for his scientific input, as well as the rest of the CHDI team, which was really beneficial for the paper, but also for ongoing work. Thank you.
Thank you very much, Joe, for that very nice talk on FAN1. Do we have any questions for Joe? Trying to see. Oh, we have here one from Patrick.
Hello. Hello. Yeah, hi, Joe. Thank you very much for this very interesting talk. I was wondering, could you comment a bit more on the therapeutic tractability of this target? What do you see there? Yeah.
So it's definitely something to seriously consider, because obviously it's a nuclease and you don't want to mess around with a nuclease. It's a structure-specific nuclease, and they're tightly regulated endogenously, so you don't want to chop up the genome. And we know that loss of function of FAN1, not that we would try and attempt that, is associated with cancer risk and genome instability. I think potentially, we really don't know enough yet, to be honest, but I would be hesitant potentially to moderate the nuclease activity directly, which is why I think this FAN1 MLH complex, modifying that might be more tractable. We obviously need to do the work for it. But we also need to understand what's the endogenous function of that complex as well. Like, it's probably not specific to resolving these CAG loop-outs and affecting efficiency there. And there's some evidence suggesting that it's it's involved in interstrand cross-link repair like FAN1 itself. But there are other tractable, that I've not specifically mentioned, there are other tractable pathways within this DNA repair network, such as MSH3 and MSH3 ASOs, which seem to be the primary candidates at the moment. But I wouldn't rule this out just yet. I think we just need to do a bit more work.
Great. Do you have any other questions for Joe? I was wondering if you could maybe speculate a bit on some of these other disorders that have been linked for susceptibility, and if you think similar mechanisms might be at play.
Well, I think the most convincing, so the genetic variation has been associated with the SCAs, the spinocerebellar ataxias and Fragile X, but a lot of the mechanistic data regarding the nuclease activity of FAN1 is really corroborated by the Fragile X model. Sorry, I'm trying to look at you through these flowers. So it's a CGG repeat expansion disorder, and they also showed that if you perform the D960A mutation, homozygous, they looked tissue specifically, they showed that that was exactly the same as a FAN1 knockout in the brain, suggesting that that is the downstream mechanism. So I didn't discuss it too much today, but that corroborates the work of Tom Massey and the entire Cardiff group, which means that the MLH1 FAN1 interaction may be upstream modifying the nuclease activity. But if you remove the nuclease activity, that's the entire function probably, in my eyes, disclaimer.
Okay. Great. I think we're, in the interest of time, we'll need to move along, but thanks again, Joe.